This is a supplemental video presentation for the CASE Animal and Plant Biotechnology course. As you view this video, be sure to make notes of important information. Feel free to pause, repeat, and resume the video whenever necessary. Sterile Technique for Plant Cloning, Lesson 4.2, Performance Enhanced Plants. Your teacher introduced Activity 4.2.2, Culturing Ferns. This video provides a review of Sterile Technique, if a review is necessary. A transfer chamber, laminar flow hood, or bacterial safety cabinet is used to minimize the potential of contaminants being introduced into the culture dishes. Nutrient-rich agar is necessary to support cell growth for tissue culture. However, the conditions are perfect to support the growth of bacteria and fungi. Bacteria has contaminated the culture in this picture. Contaminants can feed on tender plant cells and ultimately kill the culture. Other harmful effects can be from bacteria that is harmful to humans. Harmful bacteria, such as strep, can rapidly multiply in the environment meant for cell growth. Although surfaces appear clean, contaminants are everywhere on every surface. Bacterial and fungal spores can persist in the air and even the plant tissue intended to be cultured. The goal of sterilization is to destroy all contaminants. This is very difficult to accomplish, and at best we can hope to minimize the risk. The best way to sterilize is to use intense heat or pressure. Unfortunately, intense heat and pressure will also destroy the living plant tissue. Sterilization for tissue culture is primarily done by the use of alcohol-based chemicals. Be aware that clean conditions are important. If potential contamination risks are eliminated, then the risk of cultural contamination is also minimized. Sterile surfaces, instruments, and vessels maintain the sterile workspace. Personal hygiene decreases contamination from microbes found in your hair, on your skin, and in your nose and mouth. Slow, steady movements also decrease airflow over medium and sterile surfaces. If multiple groups use the same transfer chamber, members should observe sterile technique. Between steps and groups, instruments should be kept in isopropyl solution. Culture plates should be removed when you are finished. Every time someone enters the chamber, they should sanitize their gloves and the work area. Safety concerns with contaminated cultures must be paramount. Bacterial and fungal organisms could wipe out the entire population. Contaminated cultures must be removed from the growth chamber immediately and placed in an autoclave to destroy the contaminating organisms. As you review, mark or highlight 3 key points in your notes that are important to remember from this presentation. List 2 ideas or concepts that relate to previous knowledge. List any questions you have about this topic and discuss them with your teacher. Keep any notes you have from this presentation organized and available throughout the course. Now you should be ready to proceed with Activity 4.2.2, Culturing Ferns. Your teacher will provide more instructions.